Hello students, welcome back to a new video of Sunanda's tutorial. In this video we will discuss about enzymes. So we will discuss about their key properties, structural components, structural details, classification of enzymes like oxidoreductase, transferases, etc. Then mechanism of action, how enzymes work. Then models explaining the enzyme work or enzyme action, enzymatic action. Then factors affecting the activity of enzymes like temperature, pH, substrate concentration, like this. So we will discuss one by one. At first, what are enzymes? Enzymes are biocatalyst or biological catalyst. They speed up any chemical reactions without being involved or consumed. Okay. What are the main key properties? Protein nature. Most enzymes are globular proteins, though a few catalytic RNA molecule exist called riboenzymes, but mostly they are globular proteins. So they are highly efficient, extreme specificity. Each enzyme generally works on only one specific substrate for a specific reaction. So they show high specificity. Then mild condition, they function best under gentle body conditions, normal body temperature and neutral pH. Then unchanged, they help in the reaction but come out completely unchanged at the end after the formation of a product. Then structural components, many enzymes need non-protein helpers to work properly. What is apoenzyme? The inactive part or purely protein part of the enzyme. What is cofactor? It is the non-protein helper part of the enzyme. Coenzyme, which is the loosely bound organic cofactor derived from vitamins. Prosthetic group, a cofactor, okay, that means non-protein part which is tightly or permanently bound to the enzyme. Holoenzyme, it is the complete active enzyme unit. Apoenzyme, that means proteinaceous part, and cofactor, that means non-proteinaceous part, okay, which makes holoenzyme. Then classification of enzyme, there are 6 groups of enzyme. Oxidoreductase, which help in oxidation as well as reduction reaction. Transferase, help in transfer of functional groups like amino group, acetyl group, phosphate group. They can transfer the phosphate group from X to Y, like this. Hydrolase helps in the hydrolysis or addition of water reactions. Lyase help in the removal of group of atoms without hydrolysis or without the addition of water. Isomerase means rearrangement of atoms within the molecule. Ligase, joining of two molecules using energy derived from breaking down of the ATP or adenosine triphosphate. Okay, so these are the classification of different enzymes. There are 6 groups of enzymes. Then the mechanism of action. So at first, enzyme works by lowering the activation energy. What is activation energy? So reactants require certain energy, certain amount of energy to start the reaction and to start the process of converting into the product. So that is called as activation energy, and enzyme works by lowering the activation energy. In the presence of enzyme, the activation energy is required in less amount, okay, but without enzyme the activation energy is required in more amount. So in this way enzyme works by lowering the activation energy. Next, what is active site? Every enzyme has a special space or special pocket like space or cleft to add or to hold a specific substrate, and that special pocket is called as active site to attach with the substrate. Next, this site matches the exact shape and charge of the target molecule, or more specifically we can call substrate. Then substrate binding, the starting reactant known as substrate. Enzyme first bind with the substrate, then it convert it into the product. Then complex formation after attachment, the enzyme and the specific substrate form an enzyme substrate complex. Then product release, at first substrates convert into the products, then the product leave the active site, freeing the enzyme to repeat the process. It is the diagrammatic representation how substrate attach to active site of the enzyme. Then the enzyme changes the shape slightly as substrate binds, and then the substrates convert into the product. Then finally the enzyme leave that products and completely detach from that system. Okay. Next, models explaining how enzymes work. So there are two models, one is lock and key model and second one is induced fit model. Lock and key model suggests that enzymes active site and the substrate fit together perfectly like a key into the lock. So as a key fit into the lock, the enzyme fit into the substrate, so the substrate fit into the lock. And induced fit model suggests that active site is flexible and can be changed its shape slightly to hold the substrate tightly upon binding. So these are two models to explain how enzymes work or the mechanism of enzyme action. Next, what are the factors affecting enzymes activity? At first, temperature. Activity increases with temperature until it reaches an optimum temperature, approximately 37 degrees Celsius. Extreme heat changes the shape of protein and denatures the enzyme. Then enzyme stops working. Next, pH. Each enzyme functions best within the narrow optimum pH range. For example, pepsin in stomach likes an acidic pH of 2, while trypsin in intestine prefers basic pH of 8. But extreme changes in pH cause denaturation. Then substrate concentration. Adding more substrate increases the speed of the reaction, but to a certain point. After that, when all active sites are busy, the enzyme becomes saturated and the reaction reaches its maximum velocity. Okay, and finally the reaction rate decreases and finally stops. So in this way, temperature, pH, substrate concentration are three basic factors which affect the enzyme activity. Okay. So in this video I have tried to explain some basic things about enzymes. In my next video I will discuss about enzyme inhibition in details and then Michaelis-Menten equation. So please stay tuned, and if you like this video, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Thank you.